This is a production of Cornell University.
So thank you all for coming, and thanks, Bill, for the introduction. So I'm going to talk about population genetics of Phytophthora infestans. Phytophthora infestans is the causal organism of leaf blight disease, that is a disease that affects mainly potatoes and tomatoes. It can grow on leaves, stems, tubers in the case of potatoes, and fruits in the case of tomatoes. Phytophthora infestans reproduces both sexually and asexually. The sexual cycle serves, like in many other organisms, as a mode of survival and recombination. And for Phytophthora infestans to be able to reproduce sexually, it needs two different organisms. And these are termed A1 and A2 mating types. And when the A1 and the A2 mating types meet, and then they recombine their DNA material, they form an oospore, which is the sexual spore. And this is a spore that serves as a mechanism for survival because it can resist high temperatures, very low temperatures, and it can survive in the soil for many years. The asexual cycle, on the other hand, accounts for population increase, and the asexual spore are the sporangia. This sporangia can germinate directly or indirectly by releasing zoospores. And it can release up to 12 zoospores. And each of these zoospores is able to cause an infection. So whenever the pathogen is able to germinate indirectly, epidemics are much faster because instead of having one infection site, you can have up to 12 infection sites. In the United States, Phytophthora infestans reproduces mainly asexually, and that's why we have a population that is very simple, composed only of a few clonal lineages. And clonal lineages are all asexual descendants from a single individual, and variation within them arises by mutations. So how are these clonal lineages determined? They are determined utilizing a set of markers. So we have utilized an allocyte test. We determine the mating type of the individual. We do a restriction fragment length polymorphism assay using a moderately repetitive DNA probe, RG57, which can show differences between individuals. Most recently, we're using 12 microsatellite markers. We have mitochondrial haplotyping and nuclear gene sequences. In the United States, over the past 40 years, there have been approximately 24 different clonal lineages of Phytophthora infestans. But in any particular year, we only see a few of this. For example, since the late 1990s, US 8 clonal lineage is the one that has been prevalent. And this lineage is a really good pathogen of potatoes. It doesn't grow very well on tomatoes. And it's resistant to a very useful or effective fungicide called Methanoxin. And because it is resistant to Methanoxin, people stopped using this fungicide in the late 1990s. So in 2009, the US was again reminded of how devastating leaf blight is. And this is the year that, as Bill said, I came here to do my internship, so I experienced this epidemic. And why was it so bad? It was bad because tomato plants, little seedlings, were being shipped to all retail stores. So Lowe's, Walmart was selling tomato plants that were infected with the pathogen. And home gardeners and organic growers were growing these infected plants in their homes or in their fields, and they helped disperse this disease throughout the US. So in June 24, we found a professor in the department, Keith Perry, brought an infected tomato plant to our lab from one of these retail stores. And then by mid-August, it was all over New York State. So since I was here during this summer, I experienced Bill's phase of panic and doing an emergency meeting. And he invited Dr. Keith Perry and Dr. Tom Zeter to join this meeting, as well as the lab. And I saw how worried they were, and they were saying okay, what's going on? All tomatoes are dying. What's different? What's happening? And immediately I saw this was important. I just had done things in the lab, but I forgot about the impact, the real impact. So people were losing everything. They were losing a lot of money. So it was super important. So that's why I decided to come back in 2011 and figure out what was this new lineage. So as I had said before, here I'm going to show the genotypes that we have received in our lab from potato and tomatoes. And prior to 2009, US8, which is this pink color, was the dominant lineage. There were others, but this was the dominant one. Then in 2009, you can see that this red lineage, which we call US22, started displacing US8. And this US22 was that same genotype that was in the tomato seedlings that were being sold everywhere. So it makes sense that humans help disperse this disease, and that's why we see this change. But then in 2010 and 2011, we see that other new genotypes arise. These are US23 in dark blue and US24 in light blue, as well as these rare and diverse genotypes, which you can see in the striped bars, which we had no idea what they were. So I arrived in 2011, and I wanted to figure out, like, okay, what is going on with this, what's happening with Phytophthora infestans in the US. So just to put you into context, today the situation is this. US23 is displacing all of the other genotypes, and today US23 is the prevalent genotype. So again, we have 24 different lineages, but on a particular year, there's one that predominates. So my first research objective was to investigate epidemiologically important phenotypic characteristics of the recently emerged colonial lineages. These were US22, 23, and 24. And more specifically, I wanted to know what was the host preference of these genotypes, if temperature had an effect in the germination, if germination rates differed among lineages, and if this isolate differed in their sensitivity to the fungicide Methanoxin. So the first phenotype I studied was host preference, and to do this, I inoculated tomato and potato leaflets, and I placed them at 15 degrees Celsius. And after 6 days, I washed these leaflets with 3 milliliters of water, and I counted sporangia. So in this figure, what we're seeing here in the y-axis are sporangia counts. And in the x-axis, we're seeing the 4 different lineages I studied. I included US8 as a control because we already knew what we were going to expect. We knew US8 preferred to grow on potatoes. And indeed, we can see that the gray bars are potatoes, the black are tomatoes. So we can see that US8 sporulates much better on potatoes than it does on tomatoes. But we learned that US22 and US23 were very good pathogens of tomatoes. And what's even more interesting is that they were also able to grow on potatoes. And US24 was very similar to US8 in that it was able to grow better on potatoes and not that good on tomatoes. The numbers that I have within parentheses represent the number of isolates I used per genotype. So all of this, I did it in the lab, but something that was really cool was to see that when you separate that figure I showed at the beginning, but here I've only included isolates that came to the lab from potato, we can see that we see US8 and US24 like dominating. And these were the lineages that prefer to grow on potato. And then we can see that US23 is very good pathogen of potato as well. And today, like most of the samples we receive are from US23. And you see how US8 that was very important prior to 2009 is disappearing with time. On tomato, we see that US22 was exclusively found in 2009 on tomatoes. So US8, you can see we haven't gotten a sample of US8 since 2009 growing on tomatoes. There were a few isolates from US24 on tomato, but very little in comparison to those that came from potato. The next phenotype I wanted to study was the effect of temperature and germination. So as I had mentioned in the introduction, direct germination is when the sporangia forms a germ tube and it penetrates the leaf and causes an infection. Whereas in direct germination, the sporangia releases 8 zoospores, and each of these is able to germinate. So the epidemics are much more aggressive if indirect germination is happening. So how I did this experiment is I inoculated microscopic slides with water agar droplets. I put the inoculum of the I put sporangia in them, and then I placed those sporangia at 4 different temperatures, 10, 15, 20, and 25 degrees Celsius. And after 16 hours, I counted on the microscope how many sporangia were empty, indicating that those had germinated indirectly, or how many sporangia had germinated directly. So here in the continuous lines, we see the indirect germination. And what we can see is that at lower temperatures, indirect germination is favored. And as temperature decreases, as temperature increases, indirect germination decreases. Something that's also interesting is if you see at 15 degrees Celsius, US 8 and US 22 do not do that well. I mean, they don't release as many zoospores, but US 23 and US 24 do release more zoospores at 50. So temperature varies a little bit between genotypes. And in terms of direct germination, we learned it's not that common, but when it happens, it happens more at higher temperatures. So we were interested in seeing how fast are sporangia releasing zoospores, and if some genotypes release zoospores more quicker than others. So I did a similar experiment, but instead of placing the isolate at 4 different temperatures, I only placed it at a single temperature. It was 4 degrees Celsius. And what I did is counted how many sporangia were empty after 30 minutes, 90 minutes, 150, 210, and 960 minutes. And what we were amazed, because we didn't know that before, is that US 24 released zoospores by 30 minutes. 80% of their sporangia were empty. So it's really quick at releasing zoospores, whereas the other ones are slower. So we still do not know what are the implications of this, but if you can imagine a summer that has very short periods of cold temperature, genotypes, isolates belonging to genotype US 24 are going to be favored because they're going to be able to release a lot of zoospores and are going to be able to cause a lot of infection. And finally, the last phenotype I assessed was sensitivity to methanoxin. And methanoxin is a phenylamide fungicide that is widely used against oomycetes, and it's believed to inhibit the ribosomal RNA biosynthesis. This is the same fungicide that we knew US8 was resistant to, so that's why people stopped using it. To assess sensitivity to methanoxin, what we did was take isolates that had never been exposed to methanoxin, and we then transfer them to plates with no methanoxin, which were our controls, other plates with low concentrations of methanoxin, and other plates with high concentration of methanoxin. So for isolates that were resistant, basically how we assessed it is that when in our controls, there were plates that had no methanoxin when growth was 80 to 90% of the plate, then we measured the diameter of the 3 different concentrations. And we reported growth here relative to growth here. So how much less it grew here than here, or how much more it grew here than here. So US8 is resistant, and you can see that it grows very well at low concentrations and at high concentrations. But US22 is sensitive. It doesn't grow, it grows very little at low concentrations and almost nothing at high concentrations. And this is something people did not know in 2009, and that's why the epidemic was so bad. If people would have known that US22 was sensitive, they could have again used this fungicide and saved a lot of money. So we investigated this in US23 and US24 as well. And what I have in this figure is in the bottom we have the concentrations of methanoxin. The white bars represent growth at 0 with no methanoxin. So because these bars are relative to those ones, the white ones you're always going to see as 100%. And then you're going to see that US8 is resistant, whereas these 3 lineages are sensitive. Something that was very interesting is we wanted to see whether resistance or sensitivity was maintained over the years. So here we have genotypes that we collected in 2009, genotypes we collected in 2010, and genotypes we collected in 2011. And we did again the same type of study. We inoculated each of those at 5 and 100. And we can see that for US8, for example, the resistance was maintained over the years. And for US22, 23, and 24, isolates that were found in 2009 were sensitive in 2010 as well, and in 2011 as well. And like that, we saw it for US 23 and US 24. So to conclude this first part of my work, what we learned was that US 22 and US 23 were very good pathogens of both potatoes and tomatoes, whereas US 8 and US 24 were good pathogens of potato. We learned that all of the new lineages, 22, 23, and 24, were sensitive to methanoxin. We also learned that US 24 germinates much quicker than the others indirectly at lower temperatures. And this data was used by farmers to apply informed management decisions. So for example, in 2012, this blue, that kind of gray blue that we see here, is lineage US 11. So this is a lineage that we hadn't seen since early 2000, but it appeared again in 2012. And that lineage grows on tomato and potato and is resistant to methanoxin. So when the farmers sent their samples to our lab, we genotyped them, and then they receive, you have US 11 or you have US 23. So they call to our lab and they say, okay, I received that I have US 23. Should I spray or should I not? And we could tell them, yes, spray, because it's sensitive. But others that called and said, I have US 11, can we spray with methanoxin? We told them, no, don't spray because it's resistant. So people are really thankful for this data because it helped them make decisions that saved them a lot of money. So now going to my second research objective. Here I'm showing again the result for Methanoxin sensitivity. And we see that US 22 is sensitive. It grows very little at 5 and at 100. But there's some growth at 5. So we were wondering, okay, what happens if we take a plug or a sample from the isolate growing at 5 and we transfer it to 0, 5, and 100 again? So we are expecting to see this, right? But instead, this is what we saw. This is what we saw. I was going to say the results now. But, so we looked into the literature and we saw that other people had reported this, that there was some resistance after exposure to sublethal concentrations of the fungicide. So we became very interested in this phenomenon. And my second research objective was to learn the characteristics of methanoxin acquired resistance. So more specifically, I wanted to know if this phenomenon was unique to US 22 or if it was more universal for other strains. I wanted to know how fast did methanoxin acquired resistance occur, and I wanted to see the gene expression differences between initially sensitive isolates and those same isolates after they had acquired resistance. So again, I'm showing US 8. In this figure, what we can see is here, it's the previous exposure, so it means all isolates that are here have never been exposed to methanoxin before, and they were then transferred to 0, 5, and 100. Whereas isolates that are here had been previously exposed to 5, and then I transferred them to 0, 5, and 100. And we can see that for US 8, it is resistant, and after being exposed to methanoxin, it continues to be resistant. But for US 22, we see that at the beginning, isolates that have never been exposed to methanoxin are sensitive to methanoxin, but just after a single transfer through methanoxin-containing media, they increase their resistance immediately. And so we wanted to know whether this was unique for US 22 or for other isolates, and we saw that for US 23 and US 24, the same phenomenon occurred. After a single transfer through methanoxin-containing media, either low or high concentrations, resistance increased for US 23 and US 24. So, is the phenomenon of acquired resistance unique to certain strains of Phytophthora infestans? It's not. It can happen at least in 3 different originally sensitive genotypes. How fast does it occur? It occurs after a single passage through methanoxin-containing media. And now we're interested in seeing the gene expression differences between the originally sensitive isolates and those same ones with acquired resistance. So to answer this question, we used one individual of US 8, which is resistant, one individual of US 23, and 2 individuals of US 24, which are sensitive. And we grew the isolate in pea broth, and we extracted RNA from it, pea broth with no methanoxin. And then we took isolates that had already been transferred several times to 100 micrograms per milliliter of methanoxin, so they had already acquired resistance. We grew them on pea broth with 100 micrograms per milliliter of methanoxin, and then we extracted their RNA. And we sent everything to sequence, and we did 3 biological replicates. So from our RNA sequencing, we received 177 million reads, but then these were aligned to ribosomal RNA and transfer RNA to eliminate the contamination from this, and we ended up with 5.5 to 9.5 million reads. Then we aligned it to the draft genome of Phytophthora infestans, and we were left with 4.6 to 7.8 million raw counts. Then we normalized to account for the size of the genes, because bigger genes are going to have more reads than smaller genes. And after that, we studied the differential gene expression. So we were interested in knowing, these were the sensitive isolates, so we were interested in knowing the differences in gene expression between originally sensitive isolates and those same ones after a pyrocystosis. So we compared that, and we found 32 genes that were significantly differentially expressed in response to drugs in all originally sensitive isolate studies. So some of these genes were upregulated. Among these, there were 2 ATP binding cassettes, 1 phospholipase, 11 conserved hypothetical proteins, some RXLR effectors, crinklers, and endonuclease and mannitol dehydrogenase. And some of the hypothetical functions, we know the ATP cassette family are transporters, so it's logical to find transporters, because you can imagine that when you expose an isolate to stress, it wants to get rid of all these toxins that is going inside of them. And this TOMB-dependent receptor has also been implied with some antibiotic resistance. And then we saw some that were downregulated. We still do not understand very well what these genes are, how they are implicated. I mean, much more research has to be done to understand this. But in conclusion, all individuals from all sensitive clonal lineage investigated became tolerant of methanoxin. This tolerance occurred upon a single transfer through methanoxin-containing media. And among the 32 genes that were differentially expressed, there's some that could have putative functions involved in stress responses. And further work will be necessary to identify the precise mechanisms underlying this. So we have not observed acquired resistance in the field, which is good news. We saw that isolates that are sensitive have been sensitive throughout the years. We inoculated some plants, but there was a lot of variance, so we didn't really see anything in plants. This is just an in vitro observation. And we think it happens because perhaps there is a fitness cost associated with the acquired resistance, but we still need to investigate more to really understand this. So now we understand a little bit more about the prevalent lineages of Phytophthora infestans from 2009 to today. But what about these rare and diverse genotypes that happened that we saw in 2010 and 2011? So these rare and diverse genotypes were found mostly in New York State, but some individuals were found in Pennsylvania and in Ontario, Canada. And if you see in this map with the red lines, I'm showing counties where only the A1 mating type was found. With the blue lines, counties where only the A2 mating type was found. And with the squares, black squares, counties where both A1 and A2 mating types were found. So remember that when A1 and A2 mating types are together, sexual recombination can happen, and that is bad news. So I want you to look at this carefully, because it doesn't mean the fact that we are not seeing an A2 here doesn't mean there wasn't one. Our sample size was really small, so it might have been by chance that we didn't sample the A2 mating. We named these genotypes GDT1 through GDT20, and the genotypes that I'm showing underlined were those that were found in 2010, and the rest were all sampled in 2011. So much more were sampled in 2011 than in 2010. And something that I also want to highlight is that some genotypes, the ones that are in bold, were found in multiple counties. So my third research objective was to investigate the rare and diverse genotypes of Phytophthora infestans that were detected in a region around central New York. I wanted to know if these new genotypes arose either by mutation, migration, or recombination. And if they arose through recombination, if there could be any of parents among the dominant lineages that we were seeing in the US. So again, the three possible explanations for this scenario, for seeing this diversity. Remember, in 40 years, we have seen 24 different genotypes. In 2 years, we saw 20, so that's weird. So there could be three possible explanations, either mutations, migrations, or recombination. We don't think mutation is a possibility because these genotypes were diverse for their allozymes, their microsatellites, their restriction fragment length polymorphism profiles. They were polymorphic for their nuclear genes, and they showed diverse Methanoxin sensitivity. So they were different in a lot of different loci and markers. So it's really not likely that this could be explained by mutation in such a short time. So mutation, we don't think it is. But it could be migration or recombination. And migration is a possibility for sure. It could be that these genotypes arose somewhere else and they were all transported either through tubers or tomato seedlings to New York State, and that's why we found them in New York State. But to test whether they came from recombination, we did a test that is called the index of association, that is using the microsatellite loci, and this test just proves the null hypothesis of recombination, and we failed to reject it. So this genotype might represent a recombinant population. Our next question was if one of the dominant clonal lineages could be one of the possible parents. And in order to do this, and this is how it's done also for humans to see if a child is the child of some parents. So we get a strain, and then we get a potential progeny. And what we do is we look at one locus at a time. So if in that locus they do not have any alleles in common, then that's not a parent of that progeny. But if it has one allele, at least one allele in common, then it might be a parent, and then you have to go to the next locus and do the same thing. So here I have a concrete example. This is microsatellite loci. I only have 3 loci here. And what we can see is that for US8, the alleles, which in this case are numbers that are distances, sizes. For US8, it has for the D13 locus a 108 and a 112 allele. But among all these new genotypes, there were none of the alleles were either of these. So we eliminated US8 as a possible parent. And doing the same type of reasoning, we eliminated US23 and US24 as possible parents. But US22 was not eliminated. So further, looking at other markers, because that was done with microsatellites, US22 shared the same mitochondrial haplotype as the new genotypes. For the allozyme glucose-6-phosphate isomerase, US22 had a 100 and a 122 allele, and the new types had a 100, 111, and 122 allele. So this means that if US22 is a parent, the other parent had to contribute with the 111 allele. But when we look at the nuclear genes, what we found was that US22 could be a parent of some, but not of all of these new genotypes. And this is because US22 for 3 loci was homozygous for G, T, and G, whereas these 3 individuals were homozygous for a different allele. So US22 could not be the parent of these 3 alleles, but it could be the parent of the other 17 individuals. We next wanted to study population structure, and for this we included isolates that had been prevalent in the US at some point, but we haven't seen over the past 10 years. Isolates that have been prevalent in the US over this past 5 years, and the GDT isolates. And when we do this analysis, the analysis tells us that we have 2 groups. We have one group in red that comprises all of the GDT isolates, and one group in green that comprises most of the US reference isolates. What is interesting is that US22 seems more similar to the GDT than it does to any of the other US reference isolates. But because this software assumes evolutionary models that are not valid for Phytophthora infestans, then we also ran a discriminant analysis of principal component, which does not take into account any evolutionary model. And this, both the last one and this, were done with microsatellite alleles. And what we can see is that as the other test, we see the GDT group with US22, then we see the US reference isolates, but this one further differentiates US23 and US1 from the other isolates. So in conclusion, we found 20 rare and diverse genotypes composed of both A1 and A2 mating types in northeastern USA and Canada in 2010 and 11. The GDT isolates seem to be a recombinant population. It seems that US22 could be one of the possible parents for these new genotypes, but at least other two parents must exist. We don't know who they are. And if the GDT isolates represent a migrant population, it's reasonable to think that they arose at a single place and then migrated to New York State, either through tubers or tomato seedlings or humans, we don't know. So that was what we concluded for research objective 3. Now, I hope that at this point it's clear that different genotypes of Phytophthora infestans, even if they look similar, they are very different in their responses to a lot of different phenotypic traits. So they differ in their sensitivity to fungicides, they have different mating types, and they respond differently to different hosts. And this is what farmers really want to know. I mean, I don't think they care if they have US 11, 22, 23. They just want to know, should I spray, should I not? If I have tomatoes, should I be worried or not? Do I have 2 of this or just 1? That is what they want to know. So what we're currently trying to address is if there are any genetic markers associated with phenotypic traits that would allow us to determine the phenotype within 1 day or hours instead of weeks or months. That is what it takes us to do all of these experiments. So there is one problem, and the problem is that for any 2 isolates, there are millions of differences. So how do we know which of those differences are the ones that will tell us this isolate is sensitive to methanoxin, this one is resistant? So one of the ways to approach that is by doing a genome-wide association study. And to do that, we have sequenced 6 US reference genotypes, 18 GDT isolates, 36 Mexican isolates, and 1 European isolate. The Mexican isolates are very important because Mexico is believed to be the center of origin of Phytophthora infestans. So Mexican isolates are very diverse. Sexual recombination occurs commonly, from everything you sample is probably an outcome of sexual recombination. And in order to do a genome-wide association study, you need isolates that are diverse and are not linked, like the clones. And for all of these genotypes, we are phenotyping all of these traits. So today I'm just going to focus on Methanoxin sensitivity and effect of temperature on drug weight because of time, and I'll just show the results for the Mexican isolates. So again, these are Methanoxin sensitivity assays. In the top, we have growth at 5 micrograms per milliliter, low concentrations of the fungicide. And in the bottom, we have growth at high concentrations of the fungicide. And we can see that the Mexican isolates are super diverse. So if you focus on this line, this is what the control grew. The controls grew 100% because I'm showing this relative to the controls. So in the Mexican isolates, there's some that grow better in the presence of methanoxin than without methanoxin, such as these ones. And there's some that are very sensitive, that they don't grow not even like at 5 micrograms per milliliter. So this diversity is really important, and it's what we need to do this type of studies. Effect of temperature on mycelial mass. So here, remember I was saying that for germination, low temperatures favor the indirect germination, and that is good. But for mycelial growth is favored at higher temperatures. So we are interested in this phenotype because climate is changing, and we know that the summers are getting hotter, and we want to be able to predict which isolates are going to be selected when temperatures are higher or when temperatures are lower. So what we did is we transferred the isolate to 4 different plates, and we placed them at 4 different temperatures that were 10, 15, 20, and 25 degrees Celsius. And after 6 days, we measured the diameter of growth. And then what I'm presenting here is growth relative to growth at 15 degrees Celsius. So for example, we can see that isolates grow worse at 10 degrees Celsius than they do at 15, all of them. So they really do not like cold temperatures to grow. But 20 and 25, we start seeing some diversity. Some isolates grow really well at 25, better than at 15 than others, and others grow almost the same at 15 than at 25. So this is something that we think is very important, but we have to investigate. So these are the phenotypic data, but to sequence our isolates, we did genotyping by sequencing. What this does is it takes the whole DNA of the isolate, it cuts it with restriction enzymes that favor gene-dense regions, so hopefully we'll eliminate a lot of the repetitive DNA, and then we send them to sequence. So our results were, we sent 65 isolates to sequence. Most of these isolates were sent in triplicates or duplicates, so that helped us eliminate a lot of the missing data that we had. We received 570,000 SNPs after merging. And merging, I mean like with the triplicates to have one single genotype. Then we filter for proportion of missing data and minor allele frequency, and we were left with 98,000 SNPs. So remember, everything that I had shown you before was with 12 markers, 24, 36 markers. Now we have 98,000 markers. So this is important. And what's even more cool, I think, is that after doing a neighbor joining tree using those SNPs, we saw that a lot of the things that had been proven in the past were real. So that was what we were showing, like expecting to be confident on our data. So everything that is black here are isolates that came from Mexico. And what is in blue are isolates that are in the US. So US 23 is very similar to an isolate that came from Europe. And that was interesting because US 23 has also been found in Europe. So there's some connection, either Europe sent it to us or we sent it there, we don't know, but there's a connection between those isolates. US 7 and US 11 are close together. It's also very nice because there are reports that US 11 is the progeny of US 7 and US 6. Then we find that all the GDT isolates are grouped together, but what's even more interesting is that US 22 is the root of this. So it's further data to support that US 22 could be a parent of all of this, of some of these genotypes. And then we see US 8 and US 24 that are together, and they were very similar, at least phenotypically. But so after looking at this tree, something that grabbed my attention was the fact that all isolates that you see up here are able to grow on potato and tomato, whereas the ones that grow here almost exclusively grow on potato. So this is really interesting because people have tried to address this issue of host preference for a long time, and there's no clues of what's going on. But at least here we have hypotheses and we have a lot of markers that can help us understand these differences in host preferences. So this is my last slide, and these are the conclusions of all of my work. So first, what we did, we described the epidemiologically important phenotypic traits for the most prevalent genotypes in the US. We documented that Phytophthora infestans is able to acquire resistance to methanoxin in vitro, and it's important in vitro because we haven't seen this in plants. We found evidence that suggests that sexual reproduction might have occurred in the US. And this is important because right now sexual reproduction is not dominant in the US, clearly. But if it happens to become common, we're going to have to find genetic determinants to understand phenotypic traits of interest in order to make isolate-specific recommendations. And with that, I would like to acknowledge my committee members. Bill is the best advisor I could have ever dreamed of, and I'm really bad at this, I'm not going to say. And Alberto is helping me a lot with my current research, and Michael has also helped me a lot. And Chip helped me a lot with the population genetics. And yeah, thank you all. That you're please all welcome to Colombia whenever you want to come visit. This is very close to my house, and it's like that all year long. So if you ever want to have a Christmas or a nice winter break, please come visit.
So I think it's really appropriate that somebody from a very warm climate should have her thesis seminar on the winter. Are there questions for Giovanna?
So is it still the case that you only find a single clonal lineage for each farm or outbreak or whatever? Do you
Okay.
ever find two in the same place?
So the question is if from a farm, when we receive a sample from a leaf, if we find one single genotype or more than one. So this year we found at least two different genotypes in a single leaf, but that's very preliminary data. So I don't know how much we can talk about it.
Because, I mean, I guess what I'm getting at is the implications of getting a small sample from a farm and saying, yes, you can spray or not with methanoxin.
Yeah. So I think that's why we really have to pay a lot of attention in what I'm currently working on, because it's a matter of time until sexual reproduction can become dominant in the U.S. I mean, the two mating types are here. So we have been very lucky that populations reproduce asexually mainly, and because of that we have been able to manage the disease easier. But I don't know if that's going to be the case in 10 or 5 years. Yeah, and yeah, you're right. Stuart?
So do you have any information as to whether Phytophthora or seed lot plays any role in the epidemic development or the shift in the population that you've seen over the years?
So the question is if I am aware of any potatoes or seeds that might have implications in the population structure of Phytophthora that we see year by year, and I'm not aware of it. And it's hard to predict what isolates we're going to see next year, but there's a lot of explanations. For example, tubers that are left in the field and perhaps the soil didn't get as cold, so they survived, or volunteer plants that grow next year. But about like a seed source that disperses them, I'm not aware of it.
So I think we could ask Keith and Stuart if either of them knows the seed source for those potato growers in western New York.
You'd have to ask the grower. I don't know.
So first, congratulations on a really impressive body of work and an excellent seminar. My question relates to the mating. It's not prevalent in the U.S., it's prevalent in Mexico. You say it's sort of a matter of luck and it's just a matter of time before we have more mating here. But do you think it's luck, or do you think there are environmental factors that affect mating? If anybody has looked at that?
I think it's more a matter of, so the question was, why in Mexico do we see like sexual reproduction occurring frequently, and in the U.S. we do not? So a good example is Europe. In Europe, at the beginning, isolates migrated from the U.S., from Mexico to the U.S., from U.S. to Europe. And during that migration, apparently no A2 mating types went, only A1s. So in Europe, there was no sexual reproduction happening. Then there was a second migration from Mexico to Europe, and in that migration, both A1s and A2s were sent to Europe. And because there were a lot of A2 mating types, I think, it's just chance of encountering, I think. More than environmental, I think it's probability. If they survive and if they arrive and if they increase their... So places where it's 50-50 ratio have more chance of sexual reproduction than places where you see that one mating type is dominating over the other. Yeah.
If you take methanoxin tolerant variants of, let's say, US 22, and then you passage them on media without them, like with methanoxin, you get revertants.
Yeah. So we did that. So the question is, if we have the isolates with acquired resistance, and then we transfer those through methanoxin-free media, if we lose the acquired resistance. So we did that up to 3 passages through methanoxin-free media, and we didn't lose it completely, but you lose it, but not completely. So they are still resistant.
Have you tried passaging them through plants?
So we tried to do that through plants, but there was a lot of variation. I mean, like sometimes the acquired resistance isolates sometimes grew on the plants, sometimes they didn't. The ones with originally sensitive sometimes grew, sometimes didn't. So basically the answer is we don't know. We have to repeat those experiments and to know.
A related question is, did you actually, the way Alan said it, you sort of was seen implying that there was a mutation that was occurring. Do you think this is actually a mutation? That basically is selected for when you can put them on, it's one generation on this, and all of a sudden you change, or is it some sort of epigenetic kind of type modification of gene expression that's leading to the resistance?
So the question is whether the acquired resistance could be because of a mutation or because of an epigenetic mechanism. I personally think it's an epigenetic mechanism because it happens in multiple genotypes and in different individuals within genotypes. So it's highly unlikely that we get like the same typical mutations so many times and with a lot of different individuals. But I have no idea what it is, and we're trying. So I was talking in these days with Richard, that is the undergrad student that worked with me on that. There is here, Richard, and we were really interested in doing like an acquired resistant tool paper and analyzing this, because now I have DNA data as well of these genotypes. So I can make like much more accurate analysis of this.
A related question then is on the, you had, the strain 22 was the putative progenitor of all of these GTX, where these different strains were. Now you've got full genome sequence, or basically genome by, sequenced by genome, by data from throughout the genome, 98,000 SNPs. Can you go through and look at the segments within those different strains that don't match that, the 22 strain? Because then you could actually say what, because there's likely to be, a neighbor joining thing takes and crunches all the genome down into one single tree and ignores the fact that different pieces of the genome have actually different phylogenies due to the combination. Have you thought about trying to do that yet?
Okay, so the question is, if now that I have like all the data of sequences of the whole genomes and all the GDTs plus US22, if I can go over it and see like what parts of the genome are shared and which ones are not, or an evolutionary model because the tree I have here is a distance tree, so it doesn't really take into account mutations or models. So yes, that's something I've thought of, but I haven't started. Yes.
I was wondering if you wanted to speculate on why isolates become predominant and why other isolates disappear. And presumably, if you're asking this question, you have to think of where they come from. And in potatoes, presumably that would be seed sources.
Okay, so I have talked with Bill about this. He tells me sometimes that I speculate a little bit, but this is my speculation. and it's like, so if we go back to this original, this figure, we can see that sporulation is much greater on tomatoes than on potatoes. And that's why I've always thought that being a pathogen that is able to grow on tomato is really good. Like if I were a pathogen, I would want to grow on tomato because they really make a lot of spores. And potatoes, because of the tissue, it dies so quickly that the pathogen is not able to sporulate that much. So the good thing about being able to grow on a potato is that you can survive on the tuber. Because on the other hand, tomatoes don't survive very well in the winter, or not at all. But if you're able to grow on tomato and potato, then you are like the best candidate. So here, I remember when we were doing this that I had already, I told Kev, like, ooh, Kev, this US23, I said, I know it's gonna be like around for a while. And we see it today and it's everywhere. And that was something that I was just saying like to make fun because I loved doing this because there were so many spores and I liked seeing that because sometimes you never see anything like here and it's very boring. So I told that to Kev, and that was my reasoning. But again, I mean, US22 disappeared completely. It's probably because US22 we saw that it was just in a few potatoes, but it was mostly on tomatoes. And because tomatoes do not survive the winter, then it didn't have great chances of surviving. Whereas the US23 somehow happened to be in the potatoes. So it probably arrived to the US through potatoes, or I don't know. That's my speculation.
Well, if I were, oh, go ahead.
Have you or can you look for a SNP that segregates mutating type?
We have looked, and actually, so this is Alberto who's helping me do all of this, and he was like, you should have those slides. And so this is the problem with Phytophthora. Phytophthора's draft genome has like a lot of scaffolds. So Phytophthora has 8 to 12 chromosomes, but we have no idea what the order of all of these are. But if you see this, there's like a locus that seems to be promising. So this is a Manhattan plot. So what I'm looking here is scaffolds or like contigs of sequences, and then here is the inverse in the log. So the higher it is, the more significant it is. So the more markers you see that are significant is because there's something happening there that it's making an association. And here we are comparing the A1s versus the A2A. So if we eliminate all of this and just keep the bigger scaffolds, we see this. And then we saw some papers that had published. They were doing amplified fragment polymorphisms, and they had identified some loci that were present in the A1 but not in the A2. And we did a PCR in silico PCR, and we saw that it was some of the hits are in this scaffold. So I mean, this is very beginning, but we're trying to find.
Well, thank you very much, Giovanna. I think it's a really fun seminar, and are you sure you didn't set her up for that class?
I did not.
So let's thank Giovanna again for a really nice seminar.
This has been a production of Cornell University, on the web at cornell.edu.